Before we get into the experiment, we need to know a bit about phytochromes. Phytochromes are red and far-red photoreceptors which serve multiple functions at the cellular and molecular levels, as well as regulate plants' physical growth and development. They are essential for the formation of nuclear bodies, stabilizing proteins, and organizing how genomic sequences are arranged. Phytochromes allow sunlight to trigger physiological responses in plants, which optimizes their growth and fitness. Arabidopsis, the plant studied, has 5 distinct photoreceptors, phi A through E. This study focused primarily on the relationship between temperature and phytochromes, which we've already discussed, phytochrome interacting factors, or PIFs, which according to researchers at the National Taiwan University, act to repress seed germination, promote seedling development in the dark, and promote shade avoidance, and evening complex proteins, which are an important part of plant circadian rhythms.
So what exactly is their study about? Well, researchers discovered that phytochromes can in their own way detect temperatures. So in their study, they decided to investigate the effects of temperatures and the consequences that those have in regards to global warming. First, they took a model organism, Arabidopsis, which is a mustard plant, and tracked it for 8 days, ranging in temperatures from 12 degrees Celsius to 27 degrees Celsius. During this period, researchers focused on 2 things. They studied the effects of evening complex proteins, or EC proteins, as well as the effects of different phytochromes present. At the end of the 8 days, they discovered that the EC proteins ELF3 and LUX4 affected growth at lower temperatures, often decreasing the growth. However, they kind of expected these results, considering that the phytochrome interacting factor 4, which had effects on EC proteins, limited growth at low temperatures. More interestingly, though, researchers noticed that phytochrome B, or Phy-B, was particularly affected by the temperature, resulting in less growth at lower temperatures. This is important since PIF4 is regulated by phytochromes. When compared to phytochrome A, B, C, D, which had no phytochrome activity in layer O, Phy-B had the greatest amount of growth difference. Now, let's examine the data. By looking at these red and yellow data, as seen in ELF3-1 and LUX4, we can see that there is statistical significance when the plants are grown at 22 degrees Celsius and 27 degrees Celsius. The researchers also provided us with beautiful images of the plants themselves, and as you can see, the plants for both ELF3-1 and LUX4 also grew significantly taller than col 0 in general. And again, we see the same trend as represented by the data, that they are better at higher temperatures. Now let's get down to the root, or shall I say the leaves, of our data. Here is the phytochrome data, and as you can see, phytochrome B1 is the most statistically significant, and you can even see it as it grows throughout the different temperatures and how it's highest at the higher temperatures. This is what we will be focusing on in our second test.
The phytochrome Phy-B. Specifically, they compared the normal nighttime results to that of a mutant Phy-B, which locks the phytochrome in a PFR state. During the day, phytochromes will alternate between PFR and PR configurations, which exposed to far red light, or red light, at 27 degrees Celsius, the PFR configuration commonly found during the night changes into a PR configuration. These changes did not occur at 22 degrees Celsius, indicating that the changes in temperature are affecting the phytochromes. Researchers then tag and sequence Phy-B at 17 degrees Celsius and 27 degrees Celsius, recording the number of sites to which it was bound. The alternation of phytochromes at night was found to directly activate the warm temperature transcriptomes in the plant, since fewer bindings of Phy-B occurred at 27 degrees Celsius, concluding that the majority of Phy-B expression happens during the night hours. Researchers found that phytochromes like Phy-B will bind to temperature-responsive genes like PIF4. This causes PIF4 to be more sensitive to temperature changes. This sensitivity to temperature change led researchers to discover that the half-life of PFR is directly related to temperature, as the half-life of PFR is larger at lower temperatures.
So, how could increased temperature affect how phytochromes function? Since phytochromes are temperature detectors at nighttime, it's a very real possibility that global warming could affect plant function. Root respiration, which provides the energy needed for maintaining root biomass, will increase as a result of higher surface temperatures due to global warming. While this may sound like a good thing, it's actually pretty problematic. Root respiration serves as a major source of carbon dioxide loss in plants, which prevents plants from photosynthesizing. Anywhere between 8 to 52% of carbon dioxide content could be lost.